This is the second half of Chapter 5, where we're talking about chemical carcinogens. And I wanted to go through the 5 main classes of organic carcinogens. So I'm just going to go through these relatively quickly, but I did want you to be aware of what these classes were. So the first is one that I've mentioned before in regards to grilling foods, charring foods, and this is the PAHs, or the polycyclic aromatic hydrocarbons. You can see a few examples here. They all have these multi-ring structures. But again, these are chemicals that are going to be produced basically from burning any organic material, whether it be tobacco leaves or meat. The second class is the aromatic amines, or the amino-azo compounds, and these are defined by these specific chemical groups. So the aromatic amines have the amino group, usually have this, again, a multiple ring structure, and then the amino-azo compounds have that this azo group, which you can see circled here in green. And I would just point out that we already talked about one example in a little bit of detail that falls into this group, the 2-naphthalamine, which was the precursor being used to synthesize the aniline dyes and was causing cancer in that occupational setting. The third class are the N-nitroso compounds. And again, these are basically defined by their structure, having this N-nitroso group you can see here and here. And these are going to be chemicals that most people would not encounter in their day-to-day life. So this is typically going to be in terms of cancer exposure due to industrial or research activities. And next we have alkylating agents, and these are not defined based off their chemical structure. Structures can actually be pretty diverse, but are based on their ability to alkylate other compounds, including DNA, which can cause permanent mutations. We'll talk a little bit about that. One interesting example here would be mustard gas. And we'll actually come back to that because even though this compound does cause mutation, alkylates DNA, um, a lot of the early chemotherapy treatments were actually variations on this compound being used to specifically try to damage DNA and kill cancer cells. So we'll see that's kind of a contradiction that pops up a lot of some of the same things that can cause cancer may be used to treat cancer, whether they be chemicals or radiation, etc. Then we have natural products, which again is going to be broad and is just being defined based on the fact that these are compounds being produced naturally by plants. I already showed you a short list of some carcinogens that are produced by fruits and vegetables. We already talked about aflatoxin, right, the very potent carcinogen produced by aspergillus. So yeah, again, a very structurally diverse group. And then lastly, I just want to point out that there are inorganic substances that can also be considered chemical carcinogens. You can see a short list of those here, but for example, we already discussed asbestos a little bit, which is an inorganic. All right, so now I wanted to focus just a little bit on types of DNA damage that can happen in cells, how these chemical carcinogens are eventually leading to cancer, and dig a little bit into mechanism here. So a lot of these chemical carcinogens, or things that we're calling carcinogens up to this point, are technically considered pre-carcinogens, meaning the actual chemical itself is only carcinogenic if it's been metabolized, processed by the liver, and that then some metabolic product is actually the true carcinogen. But it's sort of splitting hairs, right? Because if that pre-carcinogen gets in your body converts it into a carcinogen. So we're really more interested in the initial exposure in most cases. So there's this whole process called carcinogen activation, and this should kind of remind you of the Ames test. Remember, that was the bacterial-based test that could look for chemicals that could mutate DNA. And remember, one step in that test was to mix your potential mutagen, carcinogen, with some liver homogenate to allow for this type of carcinogen activation to happen. And the main enzyme involved with carcinogen activation is called cytochrome P450. So it's a liver enzyme. There's a lot of interest in this because this is the key enzyme that metabolizes lots of drugs. So the pharmaceutical industry is very focused on how cytochrome P450 would interact with any potential drugs that are being produced. And basically what cytochrome P450 does is it's going to oxidize foreign substances in an attempt to make them more water soluble and basically easier to excrete from the body in the urine. And here's kind of a simple cartoon explanation of how cytochrome P450 would work. So you can see the enzyme here is the sort of aqua rectangle. It has this iron core to it, and it will bind the foreign substance and oxidize it. We have to bring in some oxygen for that to happen, and we'll move some electrons around. But the bottom line, what I want you to take away from this is that typically cytochrome P450 is going to be adding a hydroxyl group or even potentially multiple hydroxyl groups to the foreign substance, which is going to increase water solubility. And so some of these now oxidized compounds may be those activated carcinogens, and I'll show you an example of one of these in just a minute. But we know for sure that this enzymatic reaction is very tied in with the ability of chemical carcinogens to produce cancer. Researchers have produced knockout mice, or sorry, overexpressing mice that produce more cytochrome P450 than normal mice would, and we see they actually have an increased rate of cancer overall, suggesting that too much of this activity is further contributing to cancer development by presumably overproduction of these activated carcinogens. And so you may ask, okay, well, what is actually making these chemicals carcinogenic? And in most cases, there's a few exceptions like asbestos, but in most cases, these are going to be things that can bind to DNA and cause the DNA to mutate. So what kind of things bind to DNA? Well, chemicals that are electrophilic are going to be attracted to the very electron-rich nitrogenous bases that are part of DNA. We see a pretty linear correlation between the ability of chemicals to bind to DNA here on the y-axis and cancer-causing potential here on the x-axis. So the more something binds to DNA, the more likely it is to cause cancer. Now here's a specific example of one of these activated precarcinogens being oxidized by cytochrome P450, then binding to the DNA and causing mutation. So here we have benzopyrene, which is an example of one of the PAH class of chemical carcinogens, and it would be, again, processed in the liver by cytochrome P450. You can see where cytochrome P450 is adding multiple hydroxyl groups to that molecule. But one of these intermediates, this guy here, which has this epoxide group, it's called, is very electrophilic and will bind very tightly to nitrogenous bases, guanine in this instance. And when that happens, that binding is then going to distort the double helix. And when we get around to trying to copy the DNA, right, go through DNA replication, this DNA molecule, this is going to cause problems. DNA polymerase isn't going to know how to read that guanine, and we will likely get a base change at that location. And this image from the textbook kind of cracks me up. It's supposed to be showing sites where carcinogens can bind and affect DNA, and it's here and here and here and here and here and here and here, basically everywhere. So bottom line is lots of these carcinogens are attracted to the nitrogenous bases in particular. There's lots of places uh, where they can bind. And I also wanted to point out that there are lots of different flavors of DNA damage, so to speak, right? So from single strand breaks to double strand breaks to um, links between adjacent bases and cross links across DNA strands, etc. So there's a whole host of different types of damage that could potentially be caused by either chemical carcinogens or radiation exposure. Really, any of the things that cause cancer are typically tied in with one or more of these types of DNA damage. Um, but that being said, keep in mind that your cells do have repair pathways to try to fix DNA damage. Um, but if that is not fixed before a round of DNA replication is completed, then that may sort of lock in a permanent mutation. So let's look at this example here. Methyl nitrosourea. This is one of the N-nitroso class of chemical carcinogens that I showed you earlier. Um, this chemical can add a methyl group to guanine. And when it does that, that methylated guanine is going to want to hydrogen bond with thymine rather than the normal cytosine. So here's our initial GC pair, right, in some gene, let's say in our DNA. We're exposed to methyl nitrosourea, which adds that methyl group to the guanine. And now when we go through DNA replication, right, we split these two strands apart. The cytosine is normal, so it will be normally paired with the guanine, and we'll have a DNA molecule over here that doesn't have any mutation. But if we follow this branch, now our methylated guanine is going to be paired with thymine by DNA polymerase. And if we go through yet another round of replication, now that thymine would be paired with an adenine. So we would have gone from a GC base pair to an AT base pair here. And at this point, once we reach this situation, that's really now going to be a permanent mutation, because your cell may be able to identify this weird methylated guanine, but this is going to look like something normal, right? It's a normal base pair. So if those repair mechanisms don't catch it before this point, then we have this permanent mutation. Now, digging a little deeper into mechanism, I wanted to highlight the idea that chemical carcinogenesis is this multi-step process. So you get initiation, promotion, and then what's called progression. And so initiation is really the process of picking up mutations. Promotion is going to be exposure to really anything that is going to stimulate cell division in some way. And then we'll talk about progression here in a minute. But interestingly, a lot of studies have shown that you really need both of these events. You need initiation and promotion in order for cancer to develop. So I'm going to show you some examples where they're doing studies on rabbits, and rarely could application of a chemical carcinogen by itself cause cancer, but if you pair that carcinogen with what we call a promoting element, something that kicks on cell division, then we would see cancer rates developing and increasing pretty quickly. And also keep in mind, there could be a long gap in terms of time between the initiation event, the mutation, and this promotion event, just depending on the particular situation. But here's some of the data from these animal studies. So on the y-axis, you've got the percent of animals with cancer, basically forming tumors, and then you've got time on the x-axis. And what you see here is they were treating basically at time 0 with this chemical carcinogen. This was one of the PAH classes that we talked about. And if you just hit the, I believe rabbits in this case, with the carcinogen, little to no cancer, even over, 30 to 35 weeks. But if you pair that chemical carcinogen then with a promoter, in this case an irritant called croton oil, excuse me, and I'll show you in just a minute what this promoter is actually doing at the molecular level. Now we see cancer rates jumping way up. And that promoter by itself, if you just give that to the rabbits, right, again, basically no cancer developing. So it's that combination of the two that's really required for cancer. So what is promotion? Well, as I said, it's really anything that's going to stimulate cell proliferation. And in the case of croton oil, which is a 4-ball ester, what this chemical will do is it will directly bind to this enzyme protein kinase C in cells and basically sidestep this whole signal transduction pathway that that would normally be triggered by some external signaling molecule. Maybe a growth factor comes in, binds to this receptor, and we go through these common second messenger signaling molecules like diacylglycerol, which would then activate protein kinase C and lead to cell proliferation. But what happens is when you're hitting the rabbits with the 4-ball ester with the croton oil, we don't need that external signaling molecule anymore, right? It just directly jumpstarts protein kinase C, binds to it, and turns on cell proliferation. So again, you combine that with the exposure to the chemical carcinogen that was mutating DNA, and you end up with this situation where now these mutated cells are dividing. I'll show you an illustration of that in just a second. But do keep in mind, right, these promoters, they may be foreign chemicals, but they could also be things produced by your own body, like growth hormones, or excess growth hormone could serve as a promoting element. Things in your diet, alcohol, fat are known to be promoters, right? We already talked about the sort of the synergistic effect between cigarette smoking and alcohol consumption, right? It's basically this same idea again. The cigarette smoke would be the carcinogen, like lots of carcinogens, and the high alcohol consumption is killing cells and acting as a promoter, kicking on cell proliferation. And so then this kind of leads us into that third stage of carcinogenesis, which was progression. And just to kind of tie this all together and illustrate how you would go from some normal healthy cells to some cancerous cells. So we would have our initial initiation event, right? Exposure to some chemical carcinogen causes a mutation in the DNA in, let's say, one of these cells. Okay? And then at some point we have a promotion event that's going to trigger cell proliferation. So now the cells start dividing, and in particular, now our mutated cell has started to divide, right? So we have a bunch of cells now that contain that initial mutation, because we're just dividing, remember, by mitosis, making genetically identical copies of this cell. And then in progression, we're basically repeating the initiation and promotion stages. So we pick up additional mutation or epigenetic changes. I'll talk about these in just a second. So now, at least one of these originally mutated cells requires a second mutation. And keep in mind, for cancer to develop, these are probably going to need to be mutations in proto-oncogenes or tumor suppressor genes. But we're picking up more and more mutation, more and more epigenetic changes. Cell proliferation is continuing to be activated, or is being activated to an even higher extent. And eventually these cells are evolving into a population of cancer cells through this three-step process. So let's focus here for just a second. We're gonna see this a couple of times throughout the semester, this idea of epigenetics. And if you've taken genetics already, you may be familiar with this term. But epi means above, so this is really above genetics. And this is referring to um, genetic changes, or really structural changes that are affecting gene expression. So you're not actually changing the DNA sequence with epigenetic changes, you're changing the DNA structure, and these changes can be inherited, at least through cell division, and in some cases even from generation to generation. Um, Although a lot of these changes tend to be reset, as I'll discuss in a minute, in an embryonic setting. But common example of an epigenetic change would be methylation of cytosine bases in DNA, and excess DNA methylation can actually reduce gene expression, and we see this very typically in a lot of cancers. The methylation patterns have changed in a very dramatic way. These epigenetic changes seem to be tied in with cancer in many cases. And of course, these chemical changes, things like methylation, can be caused by chemical carcinogens, right? So that's how it really ties in with this particular chapter. So as I said, there's been a lot of interest in epigenetic changes in cancer, but I just wanted to show you one kind of interesting experiment that really kind of illustrated the fact that a lot of these epigenetic changes do get reset typically in a normal embryonic setting. And so what they did here was they removed the nucleus from some mouse melanoma cells. So you basically have a cancer cell nucleus, right, that would have presumably lots of mutations and genetic changes that would make a cell containing that nucleus cancerous, right? We already talked about the fact that it's those changes in DNA that really make a cancer cell. And they would put that that nucleus into a mouse egg cell that has had its own nucleus removed. So now you have basically a cancer cell mouse egg, essentially. And in a nutshell, what they would do is they would allow this to be fertilized and begin dividing, um, put this into a normal mouse embryo, some of these essentially cancerous stem cells. So you end up with this mouse that is essentially a chimera, where some of the cells in the adult mouse, uh, inherited the DNA from the melanoma cells, and some of the cells should be totally normal. And you might expect, okay, in this case, because a good portion of these mouse cells actually have the cancer cell DNA, that that mouse would be expected to just, drop dead immediately, or not even make it to adulthood, because it's going to develop systemic cancer. But interestingly, even though these mice do have an increased risk of cancer, they seem to be primarily normal. And what this argues is that, well, it may not just be those sequence genetic DNA changes in the cancer cells that are primarily responsible for cancer developing. It is very likely some of these epigenetic changes, the DNA methylation patterns, that were causing these original cells to be cancerous, because once we have put this nucleus now back into this embryonic environment, due to these steps that we've gone through, a lot of those epigenetic changes would have been essentially washed away. We're still left with the genetic changes in the DNA sequences, but those DNA sequence changes by themselves don't seem to be enough to cause these mice to develop cancer. So it really just speaks to the importance of epigenetics in this whole process. All right, in chapter 6, we will talk about radiation as a cause of cancer.